Good morning, everyone. Welcome to Lecture 5 of BIOC 770, where we'll be diving into the fascinating world of heat stress and how our bodies respond. I'm Mohammed Shahid Nadeem, and today we'll explore the role of proteins like HSP70 in correcting misfolded proteins under stress. Let's start with the basics. Normal human body temperature is 37 degrees Celsius. Fever or hypothermia isn't a disease itself, but a symptom. We'll discuss the different fever grades from low to high and the dangerous condition of hyperpyrexia. We'll also touch on common causes like infections and hypothyroidism. Now let's consider the opposite, hypothermia, where body temperature drops below 35 degrees Celsius. We'll examine the different levels of hypothermia and also discuss the natural variations in body temperature based on age, sex, and even emotional state. Environmental factors play a huge role. We'll identify heat sources like humidity and radiant heat. Heat stress is the imbalance between heat gain and loss. We'll explore the causes of heat stress, including dehydration, heat cramps, heat exhaustion, and the life-threatening heat stroke. Our bodies have a sophisticated thermostat, the hypothalamus. This diagram illustrates how thermal receptors send signals to the hypothalamus, triggering responses like vasoconstriction or sweating to maintain a stable core temperature. This slide focuses on the role of the hypothalamus and ADH in thermoregulation. We'll discuss ADH release, its effects on the kidneys to increase water reabsorption, and its role in vasoconstriction, particularly when body temperature drops below 36.5 degrees Celsius. Did you know the average person has 2.6 million sweat glands? We'll discuss their distribution and how sweating is crucial for thermoregulation. Evaporation of sweat cools the skin, helping us dissipate heat. Evaporation is key. We lose about 0.58 calories for every gram of water evaporated. Even without sweating, we lose a significant amount of water daily. The hypothalamus regulates sweating in response to both emotional and physical heat stress. This diagram illustrates the sweating process. Emotional stress triggers sweating in specific areas, while physical heat stress affects the entire body. The hypothalamus monitors skin and blood temperatures, initiating corrective mechanisms to maintain homeostasis. When temperatures drop, our bodies kick into action. We'll explore mechanisms to prevent heat loss, like vasoconstriction and piloerection, and to promote heat production, like shivering. Shivering is a last resort when other mechanisms are insufficient. This slide delves into non-shivering thermogenesis, particularly in brown adipose tissue. We'll discuss how uncoupling protein 1, UCP1, allows protons to leak, generating heat instead of ATP, a process crucial for hibernating animals. Heat stress can damage proteins. This slide explains how heat stress triggers the phosphorylation of heat shock transcription factors, HSFs, leading to the expression of heat shock proteins, HSPs, our cellular defense mechanism. Let's define heat shock proteins, HSPs. These are a family of proteins produced in response to stress. We'll discuss their discovery by Ferruccio Ritossa and their key functions: protein folding, refolding, and degradation. HSPs play crucial roles in stress protection, immune response, and apoptosis regulation. They're involved in host defense, protein degradation, and preventing protein aggregation. However, continuous expression can impact growth and reproduction. The temperature threshold for HSP expression varies across organisms. Antarctic organisms express HSPs at low temperatures, while thermophilic organisms require high temperatures. Even within mammals, hypothermic organs express HSPs at lower temperatures. HSPs are classified based on their molecular weight. we'll discuss HSP60, HSP70, HSP90, HSP100, and the small protein ubiquitin, each playing a specific role in cellular stress response. What's the difference between HSPs and chaperones? HSPs are a specific subset of chaperones upregulated during stress. Chaperones are a broader category, assisting in protein folding under both normal and stressful conditions. Chaperones can be classified into holduses, folduses, and disaggregases. Holduses prevent protein damage, folduses refold misfolded proteins, and disaggregases prevent protein aggregation. They act as a quality control system for proteins. Understanding protein folding is crucial. This slide illustrates the 4 levels of protein structure: primary, secondary, tertiary, and quaternary, each contributing to the protein's final functional form. Protein folding is a thermodynamic process. The free energy funnel illustrates the multiple possibilities for folding, with the native structure representing the lowest energy state. Molten globules are semi-stable folding intermediates. HSPs assist in folding newly synthesized proteins and refolding or degrading damaged proteins after stress. They are essential for maintaining protein homeostasis under challenging conditions. This diagram illustrates the complex roles of HSPs in protein folding, refolding, and degradation. They can prevent aggregation, promote refolding, or target misfolded proteins for autophagy or the unfolded protein response, UPRR. HSPs protect proteins against heat stress by stabilizing their tertiary structure, preventing aggregation, slowing muscular atrophy, and promoting longevity. They also offer protection against neurodegenerative and cardiovascular diseases. Thank you for your attention. I hope this lecture has provided a comprehensive overview of heat stress and the human body's response.